Hello everybody, Mr. Strangman here. It's time for us to go through our first lecture of the semester and investigate all of the components of Chapter 1, which is covering the scientific method and the process of scientific thought. So all of our units will be similar to this in organization. You'll have a PowerPoint screen up with my voice kind of guiding you through some of the different slides. So you'll notice that all the slides are available for you on Canvas, and you'll also be able to download those as well as some other supplementary materials. So I don't usually like to start with reading quotes during class, but in this lecture you'll have two. First, this one is by Edmund Burke, who's an Irish philosopher who once wrote, "In my course I have known and according to my measure have cooperated with great men, and I've never yet seen any plan which could not have been mended by the observation of those who were much inferior in understanding to the person who took the lead in the business." And I read that kind of fast, but what I want you to think from this is not only the old style English writing, but rather the meaning of his words. So you all have a set of life experiences and perspectives, and those are shaped by the individual life that you've led, and Burke is saying here that every perspective brings something to the table and can improve the investigation and scientific process. So keep that in mind as we move through the class. Your ideas and your perspectives could be the ones which spark the next scientific breakthrough. Now in this third slide, I want you to remember that as we move through this course, we'll be exploring both life and the scientific study of living organisms. By definition, biology is the study of life. Before we explore too much into the biology, let's start with science and scientific thinking in general. The first two images that you see are from NASA, specifically the satellite image on the right and the Mars rover on the left. Think of these images as a good starting point, as the purpose of NASA is to explore for new life. Now, here in slide 4, we're kind of looking at the start of science. And as we start, I want you to think, what does science mean to you? When I think of science, I typically think of some of those TV infomercials where they start advertising a product and say something like, "According to our scientific study, our product works 60% of the time, all the time." Well, that last part was actually a quote from the movie Anchorman, but you get my meaning. Science has become something that's less seen as infallible and authoritarian, when really science is a process of learning, thinking, and in general, gathering information. By nature, the scientific method moves us as a society forward, but within the progress, sometimes you have to take one step back in order to take two steps forward. So think of science as this massive plodding thing, slowly moving towards the idea of gaining more knowledge. Now, throughout the course, I want to give you guys some case studies to kind of analyze and critically think. And when we think scientifically, I want you to start with some kind of observation. So we'll get going with some idea or concept that really resonates with us. It makes us stop and want to learn more. So I want you to think about your major or your favorite class and what made you like it so much. For biology, I'll start here with a case study example of the scientific method and scientific thinking in action. And the example we'll use is what's called the variola virus. And this virus is better known as smallpox, and today it's actually considered to be a not very dangerous virus. We don't see much of it. In fact, it's considered to be eradicated. But in the olden days, this was considered to be one of the most debilitating and destructive pathogens that's ever faced humanity. It has resulted in deaths of millions of individuals over that time period. However, we're fortunate today to be alive in a time where the deadly disease has been eradicated. In fact, it's the only pathogen which has been considered to be eradicated from the face of the earth. So let's start with this process of how we got the destruction of a pathogen to begin. And we'll start with this man named Edward Jenner. So Edward Jenner was a fairly known and well-renowned physician who practiced medicine in England in the late 1700s and early 1800s. During his span as a doctor, he lived through some really nasty outbreaks of the smallpox virus. During one of these episodes, he was actually around a part of the country which had a large number of dairy farms. And while he was in this area, he noticed that no one on the dairy farms was contracting the disease. Being the inquisitive guy that he is, Jenner decided to investigate more closely and came to the realization that all the workers on the dairy farm had small lesions on their hand that didn't look all that dissimilar to the lesions that you would get from smallpox. So if you take a look at the image on the screen for the presentation, you'll see the presentation of smallpox infection in a child. So Jenner came to find out that the workers of these dairy farms are actually infected with a similar or related species of virus. And we call that virus the cowpox virus. So this cowpox virus caused minor symptoms but was in no way deadly like its smallpox cousin. So this paved the way for the concepts of vaccines and immunity. Jenner basically used this idea as a way to prime the immunities of high-risk individuals through infection with a non-pathogenic cowpox virus. So one observation here led us down the scientific method. Now, it's interesting to note that smallpox is the only globally eradicated disease, and the disease was officially declared eradicated in 1980 by the World Health Organization. You'll hear a lot more about them as the semester progresses. The image on the screen shows you a map of the world and highlights the dates of the last documented cases of smallpox in every continent. So in brief, North America 1952, South America 1971, Africa 1977, Europe 1953, and Asia 1975. So if we take this map information into account, we realize it actually took nearly 200 years from Jenner's initial observation until we were able to truly eradicate the disease. So science often starts small, moves kind of slow, but eventually the knowledge progresses. Now, here in slide 7, I want to take a point to note to you that every couple of slides in the lecture, we're going to take a pause to reflect and summarize some of the key points that we've covered so far. So over the first 6 slides, our take-home point and summary idea would be that science itself is a process which is focused on gaining logical, deductive knowledge to explain natural phenomenon. For your point of reflection, I would encourage you to pause the video whenever we get to these and jot down the answers to your reflection questions. Usually I'll continue straight on with the video, but I'm assuming that you're going to remember to write these answers down, as you may see these questions or ideas again, so make sure to take some time to answer the questions. I've given you an example of Jenner's observation leading to an acquisition of scientific knowledge. Can you think of 3 other examples of human processes or endeavors which are based on scientific knowledge? Why don't you go ahead and pause and write out those 3 examples here. So let's continue, and now that you've done that first bit of reflecting, I'd like to move on to our second and final quote for the term. This second quote is attributed to another renowned scientific philosopher named Karl Popper. And Popper states something kind of interesting. He says, "The history of science, like the history of all human ideas, is a history of irresponsible dreams, obstinacy, and of error. But science is one of the very few human activities, perhaps the only one, in which errors are systematically criticized and fairly often in time corrected. This is why we can say that in science we often learn from our mistakes, and why we can speak clearly and sensibly about making progress there." Now, what I hope you guys get out of that idea is that science wants you to think big and to ask large questions. Also, when you're asking these questions, errors can happen. Those errors can be associated with the original conclusion, or they can just be limited to the current set of technology which is impeding the correct conclusion from being reached. For a good example, in the 1500s, people swore the earth was flat, and there was just no way to observe the entirety of the world to disprove or even give some reason to question this. Therefore, at the time, a flat earth was considered a scientific law. Sometimes this makes me wonder what things we're doing today that'll end up being considered kind of off the wall by future generations. If you want a more recent example, in recent decades we put asbestos into a lot of different products that we sold. In fact, we put asbestos into baby clothes. And the whole reasoning was because asbestos was a flame retardant. So it made your baby sort of fire resistant. Now today we look at asbestos and we realize it's linked to all kinds of different lung conditions like lung cancer, mesothelioma, all kinds of nasty stuff. So looking back on it, was it worth putting that into the children's clothes? Probably not. Now, before we go too far into this lecture, I think we should spend some time delving into what we consider to actually be science. So as we've earlier discussed, science is a method of gaining and developing knowledge to better understand the natural world. Traditionally, as we explore the world through the lens of science, we're also looking at it through the use of the scientific method, which sounds a bit more complex and serious than it truly is. When you think scientifically, you're actually just using deductive logic to come up to a reasonable and testable conclusion, which you can repeat for validity. So science as a process is at its essence very tightly linked to social sciences like philosophy, thinking, and even reasoning, which is the crux of most social sciences. So in essence, scientific thought springs from there. Now, as we continue to move on, I've already told you a little bit that you think like a scientist consistently. You're always thinking like one, but how do you do it? So when you think or explore, you look at objects and ideas with a set of underlying assumptions. What I mean by this is, let's think, have you ever gone to see a movie and thought, there's no way that could happen in real life? Or maybe you have a friend who just can't accept when something non-believable happens in a fiction movie. That is scientifically including your assumptions about the natural world into your observations. As scientists, you'll always have some of those assumptions, such as the idea that the natural world can be understood through systematic study or a willingness to accept that all scientific ideas can be improved or revised as technology and knowledge grows. Think of this as when you write a rough draft for your term paper, and then you improve that paper and submit it for your next draft. So we're constantly working to revise. Also, you have to assume or realize that there are going to be some questions which science can't always provide a definitive answer to. So humans have this need to organize and to classify things around them. And as we make claims and statements scientifically, there's actually a hierarchy for how strong our support is for a scientific claim. So start by thinking of a cup of coffee. You can say that that coffee is cool, maybe it's lukewarm, warm, kind of hot, burning hot, etc. In science, the strongest conclusions that we can make about something is to call it a scientific law or theory. Now, as we've described earlier, these laws or theories can change over time according to new evidences. There's a phrase that I like to introduce here. It's called a paradigm shift. And it's something that's often used to say someone has changed the game or changed the way we think. This phrase was coined by another philosopher, Thomas Kuhn, when referencing the ideas of scientific discovery. So why don't you take a look at the picture on the right side of the screen? You have any ideas what that weird looking contraption is? No ideas. That is actually most of the structure of the very first microscope, and it was created by a man named Anton von Leeuwenhoek in 1666. This microscope is a prime example of an instrument which caused a paradigm shift in science. Before von Leeuwenhoek created this microscope, nobody really understood how diseases were caused or even what caused foods to spoil in the home. But von Leeuwenhoek used this tool to allow him to open up a whole new world of observation and change the way in which we see the natural world. In fact, he opened up a whole new natural world for our observation. So scientific information is everywhere now, and it's reported through a variety of modalities. And as a responsible scientist, a big part of your time will be determining what's good information and what is not so good at scientific information. In general, there are two kinds of science information. There's consensus science and frontier science. And when we're looking at consensus science, we're looking at fields in which established parameters and a wide range of published information already exists. On the other hand, when you're looking at frontier science, you're in general getting information that's less strong in its conclusions, as the information doesn't fit directly into an established parameter, and there's less supporting evidence to give a strong conclusion about the concepts. So when you're examining something, think about what kind of science it fits into. Now, I think this is another really good point for a pause, a checkpoint, and for reflection. So since we've reached our second checkpoint for the lecture, why don't we take a second to recap and answer a couple of questions? So go ahead and pause the video and take a couple of minutes to jot down your questions and your responses. First, I want you to think, what is science? How do we define it? And how do we do scientific research? So that's kind of a whole overarching question. And then, secondly, why do we say that there are some issues which are impossible for science to address? Can you think of any questions that science can't answer as it stands now? So again, go ahead and pause. I'll give a second of silence here and to give you a chance to get going, and then we'll keep moving with the lecture. So, as we move on, now that we have science established and the process of scientific thinking, it's time for us to take a look at some major theories that we'll be using to help us explore life and the human body this semester. So to start with, remember that a scientific theory is the strongest type of conclusion that we can make as a scientist based on our current level of scientific information and technologies available. The theories that we'll list here, we'll start with the first theory, and we'll call that one germ theory of disease. And the germ theory of disease simply states that every disease has a specific causative agent. And we're going to use this example through the remainder of chapter 1. The second theory that we'll explore through the course is what we call the gene theory, and it states that all organisms contain genetic information, which consists of small segments that we call genes. These genes are the functional pieces which code for an organism's characteristics. Third, we have cell theory, and cell theory is one that we'll examine in great detail in week 2 as we look at cells. Cell theory states simply that all living things are made of cells, and all cells come from pre-existing cells. The final 3 theories, homeostasis, ecosystem, and evolutionary theory, all deal with individual organisms and how they interact with the world around them. So these theories will come to be important as we move towards the end of the course this semester. To take us back to the germ theory of disease, it provides a really good starting point for us to examine how scientific theories are discovered and how they work. Specifically, it's going to use some aspects of the things we've already covered in this lecture and build on them. So remember back to the fact that Anton von Leeuwenhoek created the microscope in the 1600s. This opened up a whole world of microscopic organisms to view. However, it wasn't until the 1800s when the French scientist Louis Pasteur, have you ever heard that name somewhere else? Maybe you've heard that term, Pasteur. Think about that. Anyway, he popularized the concept of the germ theory of disease. He actually did this by discovering that a specific organism, Streptococcus pyogenes, which is actually a common bacteria of the throat and the mouth, was able to spread disease to the human body. So simply stated, the germ theory of disease states that contagious diseases are caused by germs or microorganisms which are too small to be seen with the human eye. So if you're headed down the path of healthcare and medicine, you'll be examining the role of pathogenic organisms, that is, organisms which can cause disease, to some pretty great details. So Pasteur's observations alone were not enough to create a scientific theory. Instead, the theory is based upon a body of scientific works and evidences. In the 1840s, a very famous doctor and epidemiologist, he was Hungarian, and an epidemiologist is someone who studies disease and its spread, named Ignaz Semmelweis, discovered that there was a disease, it was called puerperal fever, and it was linked specifically to women who had just given birth. So there was a high incidence of this disease in women after they'd just given birth. Semmelweis also noted that rates of infection were highest after the woman had delivered a baby and the doctor had come directly from an autopsy to the delivery room. Why do you think this would be a bad thing? Why don't you use your scientific deductive skills here? So with this, moving into slide 18, I think we've reached the halfway point through chapter 1. I think we've reached a really good point for stopping and reflecting. So why don't you take a pause from the video and answer the following questions. First, what is a scientific theory? What specifically is germ theory of disease? How was it developed? And was it one person or was it many people that contributed to this phenomenon? So pause, reflect, and then keep rolling. So as we perform science, primarily we'll be examining the scientific method. And when we're performing a scientific investigation, we're typically looking to answer questions about the natural world through a two-pronged approach, both physical observations and experimental methodologies. When we use the scientific method, we're approaching the process to use deductive reasoning. The steps of the scientific method are listed to the left side of the screen in the picture provided. And we'll go through those here. So first, when you're starting the scientific method, you want to make an observation. And that observation should lead you to a question. The question should lead you to a hypothesis or an idea of what the answer to your question is. Then you must design an experiment in order to test your hypothesis. After you've tested your hypothesis, you can collect some data, and in order to draw a strong conclusion which will determine the results which you communicate to the world, you will want to extrapolate some data. So in many cases, a conclusion of a scientific experimentation is not some final answer, but rather a process that will lead you to many subsequent questions to answer. So let's start here at the top with the first step of observation. So this is a very famous scientist. His name is Alexander Fleming, and Fleming is the scientist responsible for the discovery of the very first antibiotic, penicillin. Fleming discovered penicillin completely by accident through a chance observation. Fleming was a microbiologist, just like Mr. Strikeman, which is a scientist who focuses on the study of microorganisms. So one day Fleming made some petri plates in which he inoculated some bacteria. He then left the laboratory for a few days. Everybody needs a vacation, right? And upon his return, he noticed his plates were contaminated with mold. Specifically, it was the fungus Penicillium, which is a mold you typically see on fruits or breads in your house. What was striking about Fleming's plates was the fact that the bacteria that he had initially grown in the plate were nowhere near the spots where the contaminating mold was present. So this observation led Fleming to the question, what the heck is keeping my bacteria from growing near the mold? So to continue, once you have made an observation, often you'll need to be led to step 2 of the scientific method, asking a question. When you're asking a question, having a background set of knowledge can make it easier for you to understand what the question should be, and if it's testable. So in the slides is a really interesting link to a YouTube video from Elon Musk, and it details the questions he began to ask himself about getting around the terrible traffic that you would see down in like LA. And the video is about the creation of the Hyperloop project. So it's not a required video, but I think it's kind of a cool one to take a look at. And once you've developed your research questions, that question that you got from an observation that you're looking to answer, the next step is actually to hypothesize a possible answer to your question. So not just any question or any answer, mind you. The hypothesis should be based upon your own knowledge or research of your topic. So what does science already say about the matter? How can that lead you to a strong hypothesis? Remember that you should still have a logical hypothesis, and usually it's ideal to keep it simple so that it's easy to test, validate, or falsify. In logic, the principle of Occam's razor applies here, which is simply stating that the simplest answer is typically the true answer. So when we consider Alexander Fleming and his moldy plates, his hypothesis was that the mold, which had grown on the bacterial plates, must be creating some compound that's disrupting the growth of the bacteria. So the mold is killing those critters. Now, once you've developed a hypothesis which is reasonable, has a reasonable scientific answer, you must test it to ensure that it's valid. When you're testing a hypothesis, you must be designing some type of experiment. So put yourself in Fleming's shoes for a moment. How would you test out the hypothesis that molds can produce something that kills bacteria? As you create and perform your experiments, you often generate evidence and data which support the drawing of a conclusion. So data and evidence should be any observation that leads you to some kind of conclusion. Simply collecting data is not enough for a scientific theory or even a small-scale conclusion. A scientist must take this raw data and develop the statistics to help both summarize the data and also to draw reasonable conclusions about what it means. For example, what do your results actually say? In some of your previous courses, you may have covered the concepts such as correlation and causation. Causation basically means that one thing causes another, while correlation doesn't necessarily mean the same thing. Once you have your results, how do you effectively communicate them? Remember that every person you listen to or every article that you read is written through the lens of an author. This means that all individuals have implicit biases and ways of thinking. Good information and results produced by a scientific survey are well documented and easily repeatable. Scientists typically report their findings directly into peer-reviewed publications. Those allow for high-level information to be dispersed and reviewed by experts in the field. However, this is typically not the information which is what reaches the public. Usually scientific information is further diluted through secondary sites and sources, which turns it into more common speak. So we've now gone through the whole process of the scientific method. Let's go ahead and take a step back and return to experimentation and experimental design. Remember that an experiment's a tool to help us analyze a hypothesis. The experiment itself should exist under controlled conditions, which means it has few variables or things that could confound the answer you're generating. In these conditions, you'll typically find two types of variables. A dependent variable, which is basically referred to as an outcome variable, one that's dependent on the experiment, and an independent variable, which is one that affects the dependent variable. When we consider the design of an experiment, it's important to factor to consider is the sampling. A sample in an experiment is what is being collected or observed. Samples for experiments are typically organized as representative of a whole population. By that, what I mean is we take a percentage of the population and their results and extrapolate that to represent a whole population. For example, if you watch the news and the anchor says 60% of the population is in support of a law or a proposition, this number is a representative figure. Perhaps for this conclusion, the investigators asked 1,000 members of a population what they thought about a law, and 600 of them were in support of it. So you have to keep in mind that when we're using these samples, what's an appropriate sample size? And two things that make a sample size extra crucial are the chance for error and the experimental bias. So, chance for error and experimental bias. When we consider an experiment, another confounding factor in science is what's referred to as the placebo effect, which equates to false treatment, where you have individuals believing a sugar pill will cause them to have a health benefit, even though placebo has no effect on health whatsoever. So a placebo effect is a fake effect. We've reached our fourth and final reflection point for Chapter 1, so go ahead and pause here and take a minute to complete the following questions. First, how do experiments differ from other types of scientific observations? What makes an experiment different? Secondly, identify the concept of chance error and bias. How do those two things compare and contrast? Finally, what's the placebo effect? So I want to use the last few slides of the chapter to examine data, claims, and results. First, I'd like to address pseudoscience. Pseudo is the first example of a Latin root, which you'll see over and over again throughout the course in your future biology courses. Pseudo simply means false, so pseudoscience means false science. This is by definition a claim, belief, or practice that someone presents as scientific, but it doesn't necessarily adhere to the standards and methods of science. That is, there's no evidence testing or strong experimental design. So here are the major ways you could identify if something is science or pseudoscience. If the conclusions use vague, exaggerated, or untestable claims, if they continually say it's true without refuting evidences that state a conclusion is false, if the conclusions are closed and the data is not shown, if there's no opportunity for repeated independent experimentation, if there's no advancement of knowledge and the conclusion masquerades itself as a scientific conclusion when it's not. Finally, if the claims use misleading language or take a conclusion and information personally, it's most likely going to be pseudoscience. So in a scientific study, there are two major components. There are variables and groups. We've already looked at dependent variables and independent variables, but here we're going to be looking at an experimental variable, which is what you're physically testing in your experimentation. Every other variable in your study should ideally remain constant and unchanging. Groups are when you break up your sample population into subsets, where some are a test group, where the experimental variable is applied, and some are in the control group, which is where the group is not exposed to the experimental variable. In essence, the control group is the group which is more susceptible to the placebo effect. So as you proceed through this course, you'll be expected to examine scientific information from a variety of projects and assignments, and the best resources are peer-reviewed journal articles, which we'll work with on finding. There are websites which are good sources as well, but make sure you're critically analyzing the information you're getting and ensuring that it's the highest quality. So finally, I want to end with these two graphs. I want you to independently take a close look at these graphs and tell me what the data means. So take a few minutes to write out what you think you see here. Is there any significant conclusion from either? We'll follow up on this next week, so don't forget to view Chapter 2 as well this week. Thanks for listening and welcome to Human Biology.